The overall goal of this procedure is to observe patterns of gene expression in zebrafish embryos during specific stages of development. This is accomplished by first hybridizing a digoxygenin-labeled antisense riboprobe to the mRNA of interest. The second step is to detect the riboprobe using an antibody for digoxygenin. The antibody is conjugated to an alkaline phosphatase enzyme. The third step of the procedure is staining by the addition of NBT and BCIP, which react with the alkaline phosphatase enzyme to yield an insoluble purple precipitate. The final step of the procedure is to incubate sufficiently stained embryos in methanol to reduce non-specific staining. Ultimately, results that show gene expression patterns in whole zebrafish embryos at specific stages of development can be obtained using whole-mount in situ hybridization.
The main advantage of this technique over existing methods, like traditional classroom demonstrations of histology or dissections, is that it gives students the opportunity to build conceptual bridges between molecular biology and trends in organismal development and evolution, thereby providing a truly integrative experience.
Although these methods have traditionally been used to study the development of a few model organisms, they can easily be applied to other non-model organisms to understand the conservation of developmental pathways across vertebrate taxa, as well as where these pathways deviate, leading to evolutionary novelties.
This method may be unfamiliar to undergraduates, so some students may struggle at first. But learning this method will serve them well in undergraduate research experiences and upper division courses.
To begin this procedure, use a transfer pipette to transfer around 50 zebrafish embryos into a microfuge tube. Remove excess liquid and then pipette 1.5 milliliters of 4% paraformaldehyde into the tube. Place the tube of embryos on an agitator at room temperature overnight. The next morning, remove the fix and discard. Add 1.5 milliliters of phosphate buffered saline solution containing 0.1% tween 20 to the embryos to wash. Use fine-tipped forceps to manually decorionate the embryos if necessary. Use a transfer pipette to place the decorionated embryos into a clean microfuge tube. Then, pipette 1.5 milliliters of 25% methanol and PBST into the tube. After incubating for 1 hour, aspirate as much of this solution as possible without disturbing the embryos. Then, replace with 1.5 milliliters of 50% methanol and PBST and incubate for a minimum of 1 hour. Finally, replace with 1.5 milliliters of 100% methanol and place at -20 degrees Celsius. The embryos can be stored in methanol at -20 degrees Celsius. When ready to use, pipette the methanol from the tube and discard. Pipette 50% methanol and PBST into the tube and incubate for 10 minutes. Repeat this wash a second time. Then, aspirate the solution and repeat the incubation with 25% methanol and PBST. Finally, wash twice for 10 minutes each in 100% PBST. Prepare a 10% hydrogen peroxide solution in PBST. Immerse the zebrafish embryos in this solution at room temperature with the microfuge tube cap open for 10 to 20 minutes, depending on the age of the embryo, to remove any dark pigmentation. Using a transfer pipette, remove and discard the hydrogen peroxide solution. Rinse the embryos in 1.5 milliliters of PBST multiple times to stop the reaction. Dilute a 50 mg per milliliter stock solution of proteinase K 1 to 5000 in PBST, typically 1 microliter of proteinase K in 4.999 milliliters of PBST. Remove the PBST from the embryos using a transfer pipette. Discard and add 1.5 milliliters of proteinase K solution into the tube. Immerse the embryos in proteinase K solution at room temperature for 3 to 15 minutes depending on the age of the embryos. Wash with PBST multiple times to stop the reaction. Then, remove the PBST and immerse the embryos in 4% paraformaldehyde for 30 minutes at room temperature to refix. Remove the paraformaldehyde using a transfer pipette and discard. Add 1.5 ml of PBST to the embryos and wash for 5 minutes. Carefully pipette the PBST from the vial and discard. Repeat the wash with a further 2 volumes of PBST, then proceed to riboprobe hybridization. Pre-warm a water bath to 70 degrees Celsius and prepare the pre-hybridization solution. Then, use a transfer pipette to remove and discard the PBST. Immerse the embryos in 0.5 ml of pre-hybridization solution and incubate in the 70 degrees Celsius water bath for 2 to 3 hours. Taking care not to disturb the embryos, pipette the pre-hybridization solution from the microfuge tube and discard. Gently pipette 0.5 ml of hybridization solution into the tube. Then, quickly add previously synthesized digoxygenin-labeled riboprobe into the tube. Riboprobe concentrations ranging around 1 microgram per milliliter are used. Incubate at 70 degrees Celsius overnight. The next day, prepare solutions of 75%, 50% and 25% PHS in 2 times saline sodium citrate, as well as 2 times SSC and 0.2 times SSC solutions. Warm all solutions in the 70 degrees Celsius water bath. When the solutions have reached 70 degrees Celsius, retrieve them from the water bath. Then, aspirate the hybridization solution from the tube and discard. Add 0.5 ml of each warm PHS SSC solution, incubating the embryos in each solution for 10 minutes at 70 degrees Celsius. Finally, incubate the embryos in the 0.2 times SSC solution at 68 degrees Celsius for 30 minutes. Aspirate the 0.2 times SSC solution from the tube and discard. Incubate the embryos in 1.5 ml of malic acid buffer pH 7.5 at room temperature for 10 minutes. Repeat this wash a second time. Prepare blocking solution according to the written protocol. Transfer the embryos in MAB into each well of a 12-well plate. Remove excess MAB and pipette 1 to 2 ml of blocking solution into each well. Incubate at room temperature with agitation for at least 3 hours. Dilute the anti-digoxygenin antibody 1 to 2000 in the remaining blocking solution. Incubate at room temperature for at least 3 hours with gentle agitation. Taking care not to disturb the embryos, pipette the blocking solution from the wells of the plate and discard. Gently pipette the previously prepared anti-digoxygenin antibody in blocking solution into the wells. Incubate at 4 degrees Celsius overnight. The next day, wash the embryos in 5 changes of 1.5 ml of malic acid buffer. Allow the embryos to incubate in malic acid buffer for 5 minutes first. Then, perform buffer changes and incubate for 2 10-minute, 1 30-minute and 1 60-minute interval. Aspirate the final wash of malic acid buffer from the wells and discard. Pipette 1.5 to 2 ml of alkaline phosphatase buffer into the wells and incubate for 5 minutes. Repeat this process twice. Remove alkaline phosphatase buffer and add 1.5 to 2 ml of staining solution to the wells. Wrap the entire plate in aluminum foil to protect from light and gently agitate. At 20-minute intervals, stop the agitation and observe the embryos under the dissecting microscope to check whether sufficient staining has been achieved. Once sufficient staining is visible, pipette the staining solution from the wells and discard. Replace with PBST to stop the staining reaction. Repeat the washing process with another volume of PBST. Pipette the PBST from the wells and discard. To dehydrate the embryos, Pipette 1.5 to 2 milliliters of 25% methanol in PBST into the wells and incubate for 10 minutes. Repeat the process twice with 50% methanol in PBST for a further 10 minutes each wash. Then, transfer the embryos into 100% methanol and incubate for at least 2 hours to reduce non-specific staining. Following incubation, pipette the methanol from the wells and discard. To rehydrate the embryos, wash twice with 50% methanol in PBST, followed by 25% methanol in PBST for 10 minutes each. Then, wash in 100% PBST twice for 10 minutes each time. Finally, transfer stained embryos into 80% glycerol solution in PBST using a graded series of washes and store at 4 degrees Celsius until observation. When performed correctly, expression of the gene of interest is indicated by the appearance of a purple stain on the zebrafish embryo. This image shows a zebrafish embryo 24 hours post-fertilization, which has been hybridized with riboprobes specific for ALDH1A2. Specific staining can be seen in the eyes, hindbrain, pectoral fin bud primordia, and somites. This image shows a zebrafish embryo at the 13 somite stage of development that has been hybridized with a probe specific for FGF8A. In this case, specific staining is seen in the telencephalon, dorsal diencephalon, midbrain-hindbrain boundary, somites, and tail bud. This image shows a 22 hours post-fertilization zebrafish embryo that has been hybridized with a riboprobe specific for PAX2A, a robust marker useful for visualizing the nervous system. Specific staining can be seen in the choroid fissure, midbrain-hindbrain boundary, otic vesicle, and spinal cord neurons.
Once mastered, this technique can be modified to be performed across several laboratory sessions. Depending on the course schedule, time and effort outside of the classroom may be required by the instructor.
After watching this video, we hope that you're more comfortable with the idea of bringing whole mount in situ hybridization into an undergraduate laboratory.
As students develop these skills, knowing about this technique can facilitate their transition to upper division or graduate research experience.